Hello everyone, I'm Mary Ellen Wiltrout and I'm from the Department of Biology. So what I want to start with today is just showing you that there's a lot going on in biology, even if you only hear of the edX courses, or you may not always hear all of the other stories that happen. And today I'm mostly going to focus on 1.5 of these stories. So, but really any of these could be a separate story. So just like in physics where there's a lot going on, or other departments, there's a lot in biology as well. And just like physics, we try to use all of our MOOC content on campus as well. So our intro biology course, we've used that in 701.2, 701.3, 701.4, 701.6. And the professors either decide to use all of the content or some of the content, and they can customize things to what they want. So 701.3 has used the content for some of their own independent online problem sets, but for 701.4, they just use some of the activities in class. And also at UMass Boston, Brian White is using the content in his on-campus course. We also have a quantitative biology course. And with that content, we have used it on campus in the outreach program, because that's how it initiated from an outreach project. We also use it in the 7S 391 that we ran in 2014. We used all of the MOOC content in a blended learning environment with students that summer. And now we have that course open to everyone at MIT. If you log on to the MIT system, anyone can have access to that course for people who are in research labs and need to know this information. And our molecular biology courses, they all are used on campus as well in the residential course. But we have some projects that are not edX courses already, and we do things on campus with these courses as well. Our main project for this example would be 706 cell biology. I'm going to talk more about that later. We also did a course 7S 390, creating digital learning materials in biology. And so we worked with the MIT undergrads to make their own materials on the MITx platform, either video or problems that they wanted to do. And 703 genetics, they have used the platform and the MITx site in 2014. So we have a lot of materials, but the question is, like, are we just making all of this for no reason? You know, we really want to make sure MIT students use it. And so to first show that, I want to just show you 701.2 Introduction to Biology. And this is the activity for this semester, this year. The purple represents video events, and the orange represents problem check events. There's about 500 students in the class, and one surprising thing that you may be surprised to know is that video watching is not required at all. So we have up to 3,000 clicks on the site from students in the course, and they're not required to do any of that video watching. With the orange line, for some problem sets, they are required to go to the site to do one problem of their problem set, but only one problem of the problem set. So we also see them doing other problems on the site. They're just there for practice as well. And as you can imagine, most of these peaks align with problem set and exam deadlines. And you can see sort of in the beginning of the semester, they didn't really realize the site might be useful. And then once they got to like the end of September, they started using it. And then you can see video use is, throughout the course, even though, even outside of exam and problem sites set deadlines. So, but that's, as we expect, so. Next, I'm going to focus on the story for 706 cell biology. And we worked directly with the faculty, Frank Solomon and Adam Martin. And for this case, we wanted to really solve a problem that we were discussing with Frank during that summer, is how do we get biology students to understand experimental techniques and experimental design when they don't have experience in the lab with those techniques. And especially these advanced biology courses, they really are not doing that in the labs on campus. They're different techniques, and they want to be able to get the students to be able to discuss scientific papers, but how do we get them to discuss the scientific papers without the scaffolding they need to understand what the paper is discussing? So our potential solution we presented to Frank would be to give the students the option to learn this type of material through video. And luckily, we convinced him to give this a try. And our results at the end of the semester were that students approved and wanted more. And the professors saw an improvement in engagement and scientific thinking in the students. So I'll go through that a little bit more. So what we did is that we had 4 videos that were 5 to 8 minute animated videos. So for this project, Sarah Thornton started that summer. And so when she started, we were talking about doing these videos with Frank. What would we do? And she decided that they had to be better quality. So she taught herself animation that summer. So that's pretty amazing. And she knew how to do graphic design, but she really pushed herself to do that. And we also, on the website, we used an MITx site to host those videos. We posted questions that we wrote with the faculty to, that were directly to follow the video, that were about the video, and the students could answer those questions on the discussion forum. Frank also started using mud slips, so that's where students write the muddiest point of the lecture at the end of class if they have a question or something. He would go through those with the TAs, and the next class he would discuss one of those, the most popular one, but he would also post answers to the others on the discussion forum. And we collected data through the MITx site and surveys, and what you should also know is that all of the material on the MITx site is just voluntary. It's optional. There was no point assigned to any of the material on the site. All right. Okay, so what I'll do now is just play a clip of the video so you get a sense of what these look like.
For example, when we study signaling pathways, we want spatial information. Where is this enzyme? Where is its substrate? And we want temporal information. When does the enzyme act on its substrate? If you fix the cell to preserve geometry for microscopy, you lose the ability to assay molecules. If you open the cell to assay the enzyme in the substrate, you lose the spatial information. This video presents a neat way to address such questions. First, we'll briefly review some science from class. We discussed how phosphoinositides, for example PIP3, serve as second messengers for many signals involved in cell survival, organization, and proliferation. PIP3 is the product of the reaction catalyzed by phosphoinositol 3 kinase, or PI3K. Upstream, it is the binding of extracellular signals to the receptors that activates the PI3 kinase. What is downstream of the PI3 kinase and of PIP3? Here is the current model for one of the downstream effectors of this pathway. One of the enzymes activated by this pathway is the protein kinase AKT. AKT.
So you get the idea of what the videos are like and how they're directly tied to the class that was the material that was in content in class, as well as the material that it's setting things up for the research that was in a scientific paper. All right, and what we found when we started doing this video project is at the end of the semester, looking at all of the views, is that almost 100% of the students watched all of these videos that we posted for the course. So we have the MITx click data just showing that everyone has clicked in the video, but also just by the survey data, the students reported, about half the students that did the survey did report that they watched the video as well. And we also saw in the click data that students clicked more than just once as well. And then in the survey data, we had a lot of positive comments from students. So for example, this student said, "These videos and the discussion questions help a lot. I feel like I'm actually coming away with an understanding of the experiments and an improved ability to think about experimental design, controls, and how results can be interpreted." We also asked the faculty to, about what their experiences were. And Professor Adam Martin said, "The goal of our course is not only to get students to understand how a cell thinks, but to also understand how a cell biologist thinks. After using the videos on the MITx site, I noticed a big difference in the students' ability and willingness to suggest experiments that address the cell biological problem in lecture. They were thinking like cell biologists." So we also asked the students on the survey if they found the videos useful and the discussion forum useful. And for both cases, the majority of the students found the materials very useful as a study aid. And similar to what Safe had in his surveys, where we asked if you wanted another MITx site, if you wanted these videos and another course, the majority of students also said yes to that for our biology courses. So this year, we've added 2 additional Need Experiments videos to the course. Professor Martin has also adopted using mud slips in that practice, since when we looked at the data, we saw that even if the students weren't posting, a lot of people were using the forum to read and study. And now the course only uses an MITx site. They don't have a separate Stellar site as well. And if you look at the data for this semester so far, you can see that there's only about 100 students in this class. Purple is video, and the black line is discussion forum. And you can see the numbers we're not getting thousands of views because there's fewer students, and we have a lot fewer videos in this course. So we only have about 7 videos versus the other course, we probably have 150 video segments in the other course. But you can see that there is a regular pattern of use throughout the semester where it does go up to 200 or so events here. And what's interesting is that every day there's class. They're sort of going to the site and looking at video or looking at the discussion forum. And then, of course, when we have exams, the students are there heavily looking at the video or the discussion forum. And given that there's only 100 students in the class, having over 750 events means that they're watching it multiple times, or they're pausing in the video and like going back and reviewing things that way. And we can't really separate that out right now, but we do know from other information that the people are watching to 95% completion or more, at least the majority of students, almost everyone, 80, 90 students. So I just didn't show that graph as well. So with that, that story should hopefully be available online soon, and we can share that more. But I want to thank the faculty, Frank Solomon and Adam Martin. Of course, the MITx biology team. So Sarah's a postdoc who did the work with me on this project, and especially did all of the animation, except for one video that our UROP, Sari Riley, has done, and she's also here today. So she is amazing too with her animation skills. And we have all these other projects going on that Nathaniel, who's a postdoc, Teresa, who just graduated, she helped out some as a grad student, and now she's officially a postdoc. And Megan Warner, who just she helps out when she has time as a grad student in some of these projects. And thank you to ODL and the Department of Biology, and you should find us on Twitter and on YouTube and Facebook. And if you go to YouTube, you can see some of the other videos that we created, too, from Sari that are there, and some from Sarah. All right, thank you.